Welcome to clickmeproject.com. Let's discuss the project about automated multiscale morphometry of muscle diseases from second harmonic generation microscopy using tensor-based image processing. All chronic diseases are characterized by tissue remodeling that alters organ and cellular function through changes to normal organ architecture. We use a combined second harmonic generation microscopy and automated image processing approach to quantify morphology in an animal model of inherited MDX mouse with age. Multiphoton XYZ image stacks from tissue slices reveal vast morphological deviation in muscle from old MDX mice at different scale of cytoskeleton architecture. Cell calibers are irregular. Myofibrils within cells are twisted or larger in number compared to samples from healthy mice. In young MDX mice, such alterations are only minor. The boundary tensor approach, adapted and optimized for SHG data, is a suitable approach to allow quick quantitative morphometry in whole tissue slices. Our algorithm outperforms manual detection by time with similar reliability. Now let us see the view on the data flow diagram. This is a data flow diagram for automated multiscale morphometry of muscle diseases from second harmonic generation microscopy using tensor-based image processing. First we load our input image, and then next we construct a tensor of polar separable filters optimizing the radial part to the periodic pattern of the tissue. Due to their efficiency computation, we employ polar harmonic filters. This analysis is performed in a multi-scale framework. The energy and direction of the myofilaments represented as cylindrical tubes in the image are computed. And the next step is finding the directionality. Features can be extracted from the tensor B. Features are the energy E and the direction pi of the signals and are given by the formula given in our base paper. And the next step is phase signal. The term phase angle refers to the angular component of the polar coordinate representation. And the next step is segmentation using global thresholding. Segmentation of the filaments from the background is performed by thresholding E. And the next step is mark detector region. In this step, we will detect the abnormality. And the final step is classification using Bayesian classifier. Extracted features are trained to Bayesian classifier. Now let us see the execution of the program. First select main.m file and click run file. This is an open dialog box. This is our data set containing MDX mice, muscle diseases slices and also the human slices. We can select any one of this. Now I select this. Process is going on. This is our original image. We construct a tensor of polar separable filters optimizing the radial part to the periodic pattern of the tissue. Due to their efficient computation, we employ polar harmonic filters. This analysis is performed in a multi-scale framework. And the next one is directionality finding. Features can be extracted from the tensor B. This is our feature extraction. Here GLCM and moment invariant features are extracted. The features are successfully extracted. And then next step is phase signal. To finding the phase signal, the term phase angle refers to the angular component of the polar coordinate representation. And the next step is global thresholding. Segmentation of the filaments from background is performed by this global thresholding. And if any abnormal is detected, it returns this green color. It indicates the abnormal detection. And the final step is belonging the Bayesian classifier. This image is as a abnormal one. It returns it's an abnormal one. The multiscale analysis is presented to automatically quantify the morphometry of muscle diseases from SHG microscopy. The ongoing chronic inflammation and tissue remodeling is very well represented by the specificity of the SHG signal that maps the ultrastructural geometry within the muscle. Our approach using tissue slices also demonstrates the feasibility of imaging within tissue and the reliable quantification of ultramorphology and cytoarchitecture to map various tissue scales ranging from the multicellular bundle down to the subcellular sarcomer arrangement. Thank you.